Now that we have talked about the enzyme action, and also as you have seen in the video कि हमने देखा कि there are different factors क्योंकि everything actually depends on the active side. सारा कुछ depend in the end किस चीज पे कर रहा है? The active side. तो जाहिर सी बात है सारा focus भी हमारा उस active side पे होगा. And what are the different factors that change the active side? Because if the active side is changed in the end, what would happen? The substrate would no longer attach to the particular enzyme. So enzymes are proteins and they have a specific shape held in place by bonds. This is extremely important around the active site area as the specific shape is what ensures the substrate will fit into the active site and enable the reaction to proceed. Enzymes work fastest at their optimum temperature in the human body. The optimum temperature is 37 degrees Celsius. Now हमें पता है that the enzymes are proteins. Proteins के अंदर हमारे पास bonds होते हैं. कौन से peptide bonds? Now these peptide bonds are basically affected by temperature. Now most of the enzymes of our body work the best at 37 degrees Celsius. क्यों? क्योंकि हमारा body का temperature ही 37 degrees Celsius है. Now another key term that you need to remember in this chapter is optimum temperature. The sixth key term. हमने भी enzymes, catalyst, active sites, substrate, lock-in की hypothesis की बात की. This is the sixth term that you have to keep in mind. जिसको हम कहते हैं optimum temperature. Optimum means the best. The best temperature at which the enzymes work the best, the fastest. ठीक है और सबसे लेस टाइम कंज्यूम करके द टेम्परेचर एट विच द एंजाइम्स वर्क द फास्टेस्ट. तो जाहिर सी बात है जो हमारे ह्यूमन बॉडी के एंजाइम्स हैं they work the best at 37 degrees Celsius because they have to keep up with the human body temperature as well. क्योंकि optimum temperature of our human body is 37. सारे एंजाइम भी सबसे अच्छा काम किस पे करते हैं 37 डिग्री सेल्सियस पे. But if we start raising the temperature, heating to high temperatures beyond the optimum, अगर आप 37 से exceed कर जाएं, let's say up to 40, this will break the bonds. This will break the peptide bonds which are holding the enzyme together. Enzyme protein है. Protein कैसे बनता है? Amino acids से. Amino acids में peptide bonds होते हैं. Those bonds will break, which are holding the enzymes together, and obviously they will lose its shape. Now this is known as denaturation. इसको हम कहते हैं denaturation when the shape gets denatured. Enzyme की जो shape है वो denature हो गई. Substrates cannot fit into denatured enzyme as the shape of their active site has been lost. तो जब वो denature हो जाते हैं when you raise the temperature, they lose the shape of the active site and obviously they can no longer fit. Now denaturation is also is completely irreversible. ये एक दफा हो गया, the enzyme cannot be used again. Once the enzymes are denatured, they cannot regain their proper shape and the activity will stop. Increasing the temperature from 0 degrees Celsius to the optimum temperature increases the activity of enzymes. As the more the energy the molecules have, the faster they move and the number of collisions with the substrate molecule increases, leading to a faster rate of reaction. अब बात ये है कि yes, increasing the temperature does increase the rate of reaction, but a lot. अगर आप बहुत ज़्यादा increase कर देंगे temperature, that is harmful as it can denature the enzyme by breaking the peptide bonds. This means that low temperatures do not denature enzymes, they just make them work slowly. अब तीन scenario हैं. When temperature is low, enzymes work slow. When the temperature is low, enzymes work slow. When the temperature is optimum, the enzymes work best. When the temperature is high, the enzymes work fast. But when the temperature is too high, क्या होगा? Denature. So these are the four scenarios. ये मैं अपने students को बताता हूँ. These are the four scenarios of temperature. क्या है? Four different scenarios. When the temperature is low, वो क्या है? They work slowly. When the optimum temperature is reached, they work the best. The best temperature is the optimum temperature. हर enzyme का different होता है. When the temperature is high, when we increase the temperature, they work faster. But when we increase a lot of temperature, they would denature, they would lose their shape, no more lock and key hypothesis, and hence no reaction takes place. Look at this. This is the active site. And of course, this protein has a lot of bonds between these amino acids. सारे amino acids और proteins आपस में मिली हुई हैं with bonds. Forces between these amino acids in the chain hold the enzyme together and creates its unique shape. But once the temperature is increased, the shape of the active site has been distorted and the substrate no longer fits. high temperatures break the forces that hold the enzyme together. So this is what happening. जब आप टेम्परेचर को इंक्रीज करते हैं, obviously the enzymes start working. ठीक है, वो ज्यादा अच्छा वर्क करते हैं, and this is the point where they work the fastest. इसको हम क्या कहते हैं? The optimum temperature. ये आपका कौन सा है? This is the optimum temperature where they work the best. जब आप उससे भी ज्यादा टेम्परेचर एक्सीड कर देंगे, लाइक डबल इंक्रीज कर देंगे, फिर क्या होगा? द रिएक्शन वुड ड्रॉप टू जीरो. व्हाई? बिकॉज द एंजाइम्स हैव बिकम denatured. So this is a graphical representation. Low temperatures पे the enzymes are usually inactive. मैंने आपको बताया कि in the first scenario the enzymes are basically working slow as they are inactive. और वो आपको ग्राफ से ही पता लग रहा है. Then there's an optimum temperature जहाँ पे वो बेस्ट वर्क करेंगे. जब टेम्परेचर आप इंक्रीज कर देंगे they would get denatured. Here's another graphical representation against temperature and the rate of reaction. You can see कि जैसे जैसे मैं टेम्परेचर को इंक्रीज कर रहा हूँ, the rate is also increasing only till the point of optimum temperature. उसके बाद when the temperature is increased further up to 65 से आगे, the rate of reaction drops almost to zero. वो बिल्कुल zero पे drop कर जाता है, and hence rate is decreasing, and this means that they have lost their active site and the enzymes have denatured.